Today we will discuss about the development of the nervous system. You know that the nervous system has two main parts, the central nervous system and the peripheral nervous system. You can see that the central nervous system includes brain and the spinal cord. And when we say brain and the spinal cord, both in brain and spinal cord, which is the central part of the nervous system, you get a somatic nervous component and an autonomic nervous component. You cannot forget that, even though we say brain and spinal cord, both components are there in the same the central system. Then when it comes to peripheral nervous system, again you have peripheral nerves as cranial nerves, spinal nerves, and their ganglia. Both cranial nerves and spinal nerves have their ganglia. Spinal nerve ganglia are called dorsal root ganglia, and cranial nerve ganglia are called cranial nerve ganglia. Then the autonomic nervous system is also part of the peripheral nervous system. We have sympathetic and parasympathetic nerves, as well as their autonomic ganglia. Some are in the sympathetic chain. In relation to the sympathetic system, you get the ganglia mostly in the sympathetic chain, and in relation to the parasympathetic system, you know that the ganglia are usually close to the end organ that are supplied by them, either on the wall of the organ or close to it. So you have learned about this ganglia, I suppose. So that is the basic structure of the nervous system. In our discussion today, we will be mainly focusing on the central part, the brain and the spinal cord, but we will discuss some aspects of this the peripheral nervous system as well. Now, first you need to know about the formation of the neural tube, because the nervous system develops as a tube initially. When the three germ layers are formed, which is called gastrulation, you don't have to know all these terms, that's a different issue. In the third week, the three germ layers are set. So you get ectoderm, endoderm, and mesoderm in between. And the nervous tissue, they develop from the ectoderm, but you don't call it ectoderm. You call it neuroectoderm. And what happens is, at the back, on the posterior side, dorsal side, there is a strip of ectoderm that differentiates into what is called neuroectoderm. It's called neuroectoderm or neural plate. And that takes place immediately after the formation of the three germ layers in the third week. So the development of the nervous system also basically starts in the third week, as early as that. So it's very early. Third week is very early. Even to detect the heartbeat, actually you have to wait for 4 weeks. To detect pregnancy using a pregnancy test, you have to wait about 6 weeks. Sometimes it's a little bit early. In 5 weeks, you can this dip test. There's a chemical reaction and it gives a color then. So you know that if you check the urine of the lady, if you are suspecting pregnancy, you have to wait for 5 or 6 weeks. Usually they recommend 6 weeks. That is 36 days after missing a period. But before you do this test, you can actually, if you can get an ultrasound scan done, you can see the heartbeat at 4th week, around 4th week. Now, this is even before that. Development of the nervous system starts. There are implications when it comes to developmental anomalies. So if you want to prevent it, you have to do something very early. We'll come back to it later. But anyway, so it starts from the neuroectoderm, which is differentiated, strip of ectoderm at the back, in the dorsal side, which is called neural plate. I'll show you a diagram. Okay, so this diagram, you can see that now your embryo is like this. So this is the dorsal aspect, posterior aspect of the embryo, and this is the ectoderm that was there, and this part, middle strip, along the length of the body, middle strip, a groove is formed. Initially, it differentiates into the ectoderm differentiates into neuroectoderm, and that in the middle of the neuroectodermal area, which is called neural plate, this whole thing is neural plate. A groove is formed. A depression in the center is formed, as you can see here. That is called neural groove. Neural groove, and at the two edges, the two ends which are elevated here, they are called neural folds. Okay, so you have a neural groove and a neural fold. This groove deepens and the two folds approach each other. Ultimately, under normal circumstances, these two folds fuse with each other so that the groove becomes a tube now with a cavity inside. So that is the initial formation of the neural tube, which starts in the 3rd week of intrauterine life. And there's another thing happening during the process. The cells that were at the top of the neural fold here, the neuroectodermal cells that were at the top of the neural folds here, when the tube is formed by the fusion of the two folds, some of these cells, they fall in between the ectoderm. Now, this is no longer called, this side is no longer called neuroectoderm. It's now ectoderm. Okay. Because this ectoderm has fused with this ectoderm on either side. Okay. Neuroectoderm has gone in as neural tube. And now you get the ectoderm here. Between the ectoderm and the newly formed neural tube, some of these cells at the peak of the fold, they fall in between, and they are called neural crest cells. Okay, so these neural crest cells, later you will see they are important in the development of many structures in the body. Okay, some actually they directly give rise to, some actually without neural crest cells. If there are problems in neural crest cells, there are abnormalities in the development. For instance, anterior abdominal wall. So there are defects in the wall if you have defects in neural crest cells. So there is a long list of structures formed by the areas that develop with the help of neural crest cells. You will have to refer books to find the whole list. I'll show you some of them. Then this neural tube, if you take, now you know that there is a wall around and a cavity inside. So this cavity will be filled with the fluid later. We will not discuss it right now. Then this neural tube, we have shown you that the folds fuse and form the tube, but this fusion does not take place at once. The whole tube does not close at once. Okay. So what happens is the tube, the fusion of the two folds takes place in the cervical. I have mentioned it here. If you can read it, the fusion occurs first in the cervical region. So it starts in the future neck region, cervical region, and then the fusion spreads to the head end and the tail end, cephalic end and the caudal end. But at the head end and the tail end, an opening is left for few days. Okay, because you have to go by days, it's not a long time. It doesn't close immediately. So this hole here, the gap here is called cranial neuropore. And this gap here at the tail end is called caudal neuropore. So you can forget the name, but remember that, you know, there is a gap that is kept for some time. Actually, if you go by days, if you go to the previous slide, you can see that the head end, the cranial neuropore closes around 25 days. And the caudal end, it closes about 2 days later. So it's about 27 days. So this is how the things happen. Now the issue is sometimes this cranial neuropore or caudal neuropore can remain without closing. They can be persistent. If they are persistent, then you get what are called neural tube defects, NTD. Okay, so we'll discuss about the neural tube defects towards the end of this lecture. For the moment, remember this point. We'll come back to it later. Okay. Then few things about this neural crest cells, the cells of the top of the neural folds that fell in between the ectoderm, surface ectoderm, and the neural tube, newly formed neural tube. These neuroectodermal cells, you can see a list of structures here, which is an incomplete list of structures. You will have to complete it after reading some textbooks. So one is the melanocytes. Melanocytes are produced by the neuroectodermal cells. They migrate into the skin and they form melanocytes there. So if you have problems in neural crest cells or their migration, you will have melanocyte deficiencies or absence. Then the cells of the ganglia, peripheral ganglia. So they can be dorsal root ganglion or ganglia of cranial nerves and the autonomic ganglia. Okay, sympathetic and parasympathetic ganglia. They are all formed by neural crest cells. At the same time, you know, the adrenal gland has a cortex and a medulla, adrenal medulla is formed by neural crest cells. Okay, and then the Schwann cells. Peripheral nerves are myelinated by Schwann cells. Schwann cells are formed by neural crest cells. And these are only some of the structures that develop from the neural crest cells. The list can be extended, and there are there are things that does not develop from neural crest cells, but for the complete development, proper development, you need the assistance from neural crest cells. You find out an embryology book can complete your list. Okay, so these things sometimes come in MCQs, true, false MCQs and all. Okay. Okay, when it comes to the main part of the lecture, the development, now we will concentrate a little bit on that. When it comes to the development, initially what happens is the neural tube that was formed expands at the head end, cephalic end. It expands there and forms three vesicles. Okay, it forms three vesicles. The first vesicle is called forebrain vesicle. Now these terms, prosencephalon, mesencephalon, brombencephalon, if you can remember easily, you can remember, no problem. But I also forget, some prosencephalon I remember, but other things I forget. Okay, but it's easy to remember them as forebrain, midbrain, and hindbrain. Okay, so you have forebrain vesicle, you have a midbrain vesicle, and the third one is the hindbrain vesicle. So initially, this tube expands to form three vesicles like this. Okay, then later, after some time, few days after, these vesicles, their growth is different. Some vesicles they grow very rapidly, others they don't grow very rapidly, and there are differences made. Now the first vesicle, the forebrain vesicle, as you can see here, divides into two further parts. I have labeled them as 1A and 1B. These are not standard labeling. Okay, so there's nothing like 1A, 1B in textbooks. Okay, it's only in my lecture. Okay, but just remember the point that the forebrain vesicle divides into two parts: anterior part, cephalic part, and a caudal part. Then the second one, the midbrain vesicle, as you can see here, it remains as it is. Okay, it doesn't divide or enlarge too much. It remains as it is. Throughout, it's like that. It remains as it is. Okay, and the third one, hindbrain vesicle, again it divides into two parts. I have labeled them as 3A and 3B. Okay, then you can also see that the first part of the forebrain vesicle grows rapidly and expands and hides the second part of the forebrain vesicle. Okay, so the first part grows rapidly and the second part gets hidden inside the first part. Okay, later when we label, if you look at an adult brain and you label the cerebral hemisphere, the parts of the brain, you actually label this 1A, the first part of the forebrain vesicle that was growing rapidly, you label it as cerebral hemisphere. So in the adult brain, you have left and right cerebral hemispheres. And this small part of the forebrain vesicle, the second part of it, that is called diencephalon. I don't know whether you use this term when you learn the structure. Again, you don't have to remember all this telencephalon, diencephalon, metencephalon, myelencephalon. Okay, so this is for technical purposes, educational purposes we have put. Just remember that the second part, it actually consists of the thalamus and hypothalamus mainly. Okay, for the completeness, I have added the other two also, subthalamus and epithalamus. Okay, but the two main ones are the thalamus and hypothalamus. You can forget the other two and remember thalamus and hypothalamus. Since you need some sort of a common name for both thalamus and hypothalamus, we can, if you can remember it as diencephalon, even though it may be difficult for you. Okay, so you have the cerebral hemispheres and the diencephalon consisting of thalamus and hypothalamus. And in the adult brain, if you cut the brain, you will see that these two are hidden inside the cerebral hemispheres, left and right cerebral hemispheres. So if you want to see them, actually, you can't see them from the lateral view. So if you look from here, where the laser pointer is, you cannot see them because it's hidden by the lateral side of the cerebral hemisphere. For you to see them, either you have to cut the cerebral hemisphere here and remove it, so that you will see that side of the diencephalon, or you have to take either you can cut this side and see that side, or you take a sagittal section, mid sagittal section here, then you will see the the sagittal section of the diencephalon. Okay, either you can cut it and see, or you can remove the cerebral hemisphere on that side and see the lateral side of it. I think you can understand it. Then, as I said before, the midbrain, the midbrain vesicle, the same name. Okay, midbrain vesicle here gives rise to the midbrain that you have learned in your structure. Midbrain. Okay, so midbrain has its parts. I'm not going into details of that. Tectum, tegmentum, and cerebral peduncle, crus cerebri. Then the third one, the hindbrain vesicle, which got divided into two further vesicles. The first one, upper one, gives rise to pons, upper medulla, and cerebellum. Pons, upper medulla, and cerebellum. Then the lower one gives rise to the lower medulla. Okay, so the only difference is that they don't have any other important things to remember. So anyway, it's 1, 2, 3, 4, 5 things that develop from these things. Certain things you can remember, but certain things it's not very important to remember. But for the completeness we will learn. Then the cavity that was inside the original neural tube, it will not stay as a tube cavity in a tube. That will also expand with the expansion of the tube. So you can see that the cavity that was inside the anterior end of the neural tube now expands with the developing cerebral hemispheres, and that has given rise to two large ventricles. They are called ventricles because they are large cavities. And so you have the left and right lateral ventricles in the two cerebral hemispheres. And they are connected up by a foramen, because they were one cavity actually initially. So they are still, they maintain their connection through foramen of Monro. Through foramen of Monro, they are connected. Then the cavity that was inside the diencephalon, thalamus hypothalamus area, that expands and gives rise to the third ventricle. Then the cavity that was inside the midbrain vesicle, which did not change much, it remains as a small tube, which is called cerebral aqueduct. Cerebral aqueduct. Again, the cavity that was inside the pons, upper medulla, and cerebellum area, that also expands and gives rise to the fourth ventricle. And then the cavity inside the lower medulla and the spinal cord that is below that remains without much change as the central canal of the spinal cord. So this is the structure that you actually see when you study the brain. And all these things are connected, interconnected, because it's one single cavity that expanded. Okay, so the connection will not lose. And later you will learn that the fluid that is inside will be actually produced in these lateral ventricles and in the third ventricle, and they actually they travel, they pass through the tube, and they come out through the fourth ventricle, roof of the fourth ventricle, and they circulate around the spinal cord and the brain, the subarachnoid space, before they ultimately get absorbed into the superior sagittal sinus through the arachnoid granulation. So here the structure that produce the fluid is called choroid plexus, and the structures that absorb the fluid into the bloodstream, superior sagittal sinus mainly, they are called arachnoid granulations. That much I think. Okay. Okay, then when it comes to the development of the spinal cord, this part, development of the spinal cord, this is how it happens. Now, initially you have this neural tube, which is formed by neuroepithelial cells. You can call them neural stem cells because they are going to give rise to all the other neural cells. And inside you have this cavity. Okay, this is basically the initial neural tube. Now, this one, the wall, the neuroepithelial cells, they proliferate rapidly and they give rise to two layers, one after the other. Now the tube wall is very thick, give rise to two layers, and they further develop, and then you actually name these two layers. Again, you don't have to remember the names as such. The layer that is inside is called mantle layer, and the layer outside is called marginal layer. Terms are not very important. Inside, the neuroepithelial cell layer remains because it still develops. So the same picture, I took it here. Then the other important thing is, after some time, after a few days, you don't have to remember the exact date, a groove develops in the center. Okay, in the center means if you have a posterior half and an anterior half, between the posterior and anterior halves, a groove developed, which is called, grooves are called sulci. It's called sulcus limitus. So sulcus limitus indicates an anterior half and a posterior half in this developing neural tube where the spinal cord is going to develop. And the anterior half here is called basal plate and the posterior half is called alar plate. You can forget these terms and you can remember it as motor plate and sensory plate. Okay. Anterior half is the motor plate and the posterior half is the sensory plate. Now later when the neurons develop, nerve cells develop, all motor cells develop in the anterior half. All sensory cells develop in the posterior half, whether it's somatic or visceral, somatic or autonomic. The sensory ones develop in the posterior half, motor ones develop in the anterior half. So that should be clear. Okay, now in the red circle indicates motor cells, blue circle indicates sensory cells. So there's another circle. Now this large red circle represents somatic motor cells. Small red circle represents autonomic motor cells. Then the large blue circle represents somatic sensory cells, and the small blue or green one represents autonomic sensory cells. Okay, so the first rule, sensory half and motor half, it does not change. Okay, and you can see another thing, that the somatic ones, they are further away from the sulcus limitus, the plate, the line passing through the sulcus limitus, so the plane passing through the sulcus limitus. Somatic ones are further away from that plane, and the autonomic ones are on either side, in front and behind of that plane passing through the sulcus limitus. Okay, so this is a general rule for the spinal cord. Then these neural crest cells, as I told you before, they migrate from where they were produced initially, and they come and form the dorsal root ganglion in relation to the posterior half of the spinal cord. So this is the same diagram. I have made it small to compare it with another one. So you know the posterior half is sensory, anterior half is motor. And if you compare it with the structure that you have learned when you learn the spinal cord, you know this spinal cord, by looking at it, you know that it's a posterior horn, lateral horn, and anterior horn. Most probably it's a thoracic spinal segment section that has been drawn here. And the same rule remains: posterior half is sensory, anterior half is motor. So you get your sensory cells from that side coming in, having the dorsal root ganglion outside the central nervous system in the intervertebral foramen area, then the nerve fiber entering the posterior horn. And you know, it can either ascend up, it can have, you know, connecting neurons, and the secondary neuron can connect up, or it can, you know, interneurons with, it can connect up with the motor, somatic motor neurons for reflexes. So you know this stuff, right? Then the sensory nerves also, say autonomic nerves, sensory autonomic nerves also, visceral nerves, come through this path. They have their cell bodies in the dorsal root ganglion and enter the posterior horn, and they will have their sensory cell bodies in that area, okay, interneurons or whatever, and the motor cell bodies in front of the plane. And then they also leave with these same nerves, because all these, your posterior nerve root and anterior nerve root, they will be pure in the sense that you have a sensory root and a motor root in front and behind, but they are not pure in the sense that they have somatic and autonomic ones mixed. Okay, when it comes to the actual spinal nerve, once the two nerve roots, I don't know whether you understand when I draw it like this, posterior nerve root and anterior nerve root, they unite inside the intervertebral foramen and form a spinal nerve that is mixed in all aspects, somatic and autonomic, as well as sensory and motor. Okay, so these are things that you should understand properly. Then when it comes to the development of the brain stem, again, you have learned the development of the brain stem. This basic structure of the neural tube that breaks at the back, posterior end, it breaks and opens up like this. Breaks and opens up like this in relation to, especially in relation to the fourth ventricle. When I say fourth ventricle, you get, that is the pons area and the upper medulla area, pons and upper medulla area. So this opens up like this. So even, you know, even though there is no expansion, you get the same arrangement here also. But basically, it's in the fourth ventricle. So this is the flow of the fourth ventricle which you see here, flow of the fourth ventricle. Now imagine when this happens, when this breaks and opens up like this, the somatic motor neurons that were there. I'm referring to the cell bodies. Okay, neurons are the fibers are outside. Somatic motor cell bodies that were in front here. Now, because of this opening up, if you take this midline, this is the midline now, they are closer to the midline. These ones are here now. And this is the area of the sulcus limitans. Okay. The autonomic motor ones, visceral motor ones are close to the sulcus limitans but medial. This one. Okay. And the visceral sensory ones just lateral to it. Okay, visceral sensory, autonomic sensory. Then these somatic sensory ones are further away. Somatic sensory ones. So this general arrangement, when you learn the cranial nerve nuclei, cranial nerves have their cell bodies in the brainstem. They are called nuclei. This is the usual arrangement of nuclei in the cranial nerve. This does not stay like this, in all places. There are certain changes. Things move here and there during development, but you need not get confused. You should remember the basic rule, that's all. Then when it comes to the head and neck region, other than in these sensory, sensory and motor components that they are in the spinal cord, when it comes to the head and neck region, you get an additional component here in between, which is called branchial motor component, which I have omitted, intentionally to avoid confusion. But there's a third part coming in here. Okay, if it is difficult, you don't take it very seriously. But I will give you a few points about it towards the end of the lecture. Then this concept of somites and spinal segments, which is an important concept. Now, even though we don't see our bodies as segmented things, you know that if you look at a creature like earthworm, or even there are other creatures who get like earthworms, they get segmented bodies. But in our setup, we don't see this segmentation very clearly outside, except when you look at your spine, you know that it is segmented because there are vertebrae. There are 33 vertebrae, some are fused in the caudal end, but you obviously see the segmented nature in the vertebral column. And again, when it comes to the thoracic region, you get the ribs and intervertebral, you get the intercostal muscles and ribs. Again, you see the clear segmented nature, but this only either you have to touch it and see, or you have to remove the skin and the muscles to see it. Otherwise, you don't see it. But during the development, this segmentation, like in lower animals, we get this full segmented development, at least in relation to the neural tube and the vertebral column area. So these segments that are formed in relation to the neural tube in the center, they are called somites. Okay, they are called somites. You can see so many somites here. Some are in the neck region, some are in the thoracic and below. So that's an important point to remember. Now these somites develop from what is called paraxial mesoderm. Now again, you must have learned about the classification about the mesoderm. You have the mesoderm that is on either side of the developing neural tube, that is called paraxial mesoderm. Then mesoderm that is in front, that later forms the trunk and all, that is called lateral plate mesoderm. Okay, and the mesoderm in between, the paraxial mesoderm and the lateral plate mesoderm, here is called intermediate mesoderm. Okay, now this paraxial mesoderm that is on the side of the neural tube, that will actually give rise to certain parts of the skin of the body and muscles of the body and vertebrae that surround the neural tube and later forms the vertebrae by the paraxial mesoderm. And therefore these somites that are developing, they are actually formed by the paraxial mesoderm. Okay, so this has to be meshed with this part. Then I will tell you a few more things. This intermediate mesoderm is the one that gives rise to your gonads and kidneys. Okay, gonads and kidneys. So it's genitourinary systems. And then this lateral plate mesoderm will give rise to the body wall musculature and certain structures like ribs, I think. And then you get the mesodermal structures like smooth muscles surrounding the tubes inside the gut and respiratory tract, all that by the visceral part. So you have a parietal part of the lateral plate mesoderm and the visceral part. So I just summarized it to show you the relevance. Then these somites that were there that developed from paraxial mesoderm, you see here, somites they further develop into three areas. There's a sclerotome area in the somite, there's a dermatome area, and a myotome area. And this the spinal cord that is developing inside, even though it is not divided into areas with lines, you see it as a tube. But when it comes to functional aspects and when it comes to the nerves that are coming out, spinal nerves, they have a series of spinal nerves. Each spinal nerve is coming out from a spinal segment. So in relation to each somite, the tube gets segmented. Okay, so you get fifth somite, you get fifth spinal cord segment, like that. Okay, so then what happens is this somite that got separated into a sclerotome, myotome, and dermatome. What happens? The sclerotome will, the two sides will unite here, and later the posteriorly also the cells migrate, and you can see some cells migrating and form the, you get the vertebral arch here, you get the vertebral body here, and the transverse process and the spine. So it forms the vertebra in relation to this spinal segment. Okay, this spinal segment, the vertebra is formed by the sclerotome. Then the dermatome will form an area of skin supplied by the spinal nerve arising from the spinal cord segment. And the myotome will give rise to some muscles, some muscle tissue in the body supplied by the spinal nerve, motor component of the spinal nerve arising from the same spinal nerve coming from the spinal segment. Okay, so this is the sensory component for the dermatome, and this is the motor component for the myotome, for contraction of muscles. So this is an important thing to remember. Okay, so this I mentioned. Then this, if you by now you know what is a dermatome. Dermatome is an area of skin supplied by a single spinal nerve or spinal cord segment. Okay, so that is sensory. So you have learned about this dermatomal maps in the trunk, upper limb, lower limb, and head and neck region. So say if you take T2 dermatome, that actually supplied by the T2 spinal nerve coming from the T2 spinal cord segment. Okay, because of this somite developmental theory. Then if you take myotomes, you define it as an area of muscle in the trunk or limb. You can drop that area of muscle supplied by a single spinal nerve, spinal cord segment, the same thing. This is sensory, this is motor. And if you have learned upper limb now, if you take biceps muscle, we say that it is supplied by C5 and C6. I don't know whether they have mentioned it here. So they have mentioned only one, but it is supplied by both C5 and C6. So it has two dermatomes, C5 and C6 dermatomes in the biceps muscle. So it actually, that supply should come from two segments there. Okay, from contribution by two spinal nerves. Later, in the brachial plexus, they combine and form one nerve. That's a different issue. So they come through one single nerve, but origin is from two spinal nerves coming from two spinal segments. So then this is the principle behind the development of these things. Then when it comes to the head and neck region, this concept of branchial arches and cranial nerves, which you have not learned, because I can understand sometimes students get confused because of that. Teachers can drop this concept altogether. But I think at least for few students, it may be important if you want to read and understand textbooks. So I will give you a very, very superficial count of the branchial arches. Now, when again, like, we get our body segmented, at least during a certain period of development, just like that, in our pharynx area, the neck region, the pharyngeal region, we get a formation of gills. Gills, fish have got gills, karana. So you get the development of gills, gill-like structures in our neck region. So they have these areas, they have these arches actually, and in between there are clefts. You can see here arch and cleft. Cleft areas are like gills. But after few weeks, I think few weeks or days, the clefts close up. They don't remain like in fish. They close up, and you get your development of the pharynx. But you do get problems with this closure of clefts. Sometimes you leave one or two behind, giving rise to branchial cysts and all that. Okay, branchial fistula, branchial cysts. You will learn it when you do the surgery appointments. Okay, so that for that reason you should know about branchial arches. Okay, otherwise if you skip it, then you will have problem in explaining these clinical cases. But my point here is not the branchial cysts or sinuses, but my point here is that when it comes to development of the nervous system, what is important to know is that the branchial arches that are developing during some stage of development in the pharynx area, they give rise to many structures in head and neck region. Okay, many structures in the head and neck region. If you take a single branchial arch, say the first branchial arch here, you can see there's a tissue inside in pink color. Then there's a lining here and lining there, outside and inside lining. So inside lining is endoderm, you know that. Outside lining is ectoderm, and in the middle you get the mesodermal core. Okay. Now this endoderm will give rise to certain structures in certain places. Ectoderm will give rise to certain structures, and the mesoderm will give rise to certain structures. So what are the mesodermal structures? You get either bones or cartilages or muscles. Bones, cartilages, or muscles. And the important thing is structures that develop from this, we will say first, forget about the endoderm and ectoderm. If you take the mesodermal core, structures developing from the mesodermal core, especially the muscles, they are supplied by arch nerve and arch artery. Okay, so there is a specific arch nerve that supplies structures developing from that. Specific arch artery that supplies structures developing from that. We are not concentrating very much on the artery anyway, We especially go by the nerves because nerves remain without much change. So I have mentioned it here, each arch will have its own bone, cartilage, muscle, nerve, and artery. Now you can see here, these structures like, some middle ear ossicles like malleus and incus and the maxilla, mandible, these things and muscles of mastication, they develop from the first pharyngeal arch mesoderm. First pharyngeal arch mesoderm, and the first pharyngeal arch nerve is the mandibular division of the trigeminal nerve. Therefore, those muscles of mastication, they are supplied by the mandibular division of the trigeminal nerve. Then these bones like stapes, middle ear ossicle, then the styloid process, stylomandibular ligament, and lesser horn of the hind. I mean, that part is not very important, but then the muscles of facial expression. All these develop from the mesodermal core of the second pharyngeal arch, and the arch nerve is facial nerve. Therefore, all these things are supplied by the facial nerve, especially the muscles of facial expression and rhetoric. Okay. Then the pharyngeal muscles, they develop from the third arch. Pharyngeal muscles develop from the third arch. Third arch nerve is the glossopharyngeal nerve. So they are supplied by the glossopharyngeal nerve. And some of the pharyngeal muscles again, and some laryngeal muscles, they develop from the fourth pharyngeal arch mesoderm. Fourth pharyngeal arch nerve is the superior laryngeal branch of the vagus nerve. Therefore, laryngeal muscles and some of the laryngeal muscles and the pharyngeal muscles are supplied by the vagus nerve as well. Then the recurrent laryngeal nerve, that also, that is the sixth arch nerve. Fifth arch disappears later. Sixth arch nerve, the structures developing of the sixth arch nerve are parts of the larynx and the laryngeal muscles that is supplied by the recurrent laryngeal nerve with sixth arch nerve. Okay, so vagus nerve has two parts here. So if it is difficult to remember, you can just remember which nerve supplies which muscles, but this is the basis of that. Okay. Then there's another important point. This is more important than the previous one that I mentioned. Okay, you can't do this one. This is more important than the previous one. Now, when the vertebral column develops from the sclerotome of the somite and the spinal, the neural tube develops into a spinal cord, the growth does not, the speed of growth does not match. Speed of growth of the vertebral column does not match with the speed of growth of the spinal cord. Now, first you try to understand this diagram, this is a very diagrammatic thing. So these things represent the vertebral vertebrae. T12 to L5 vertebrae are drawn here, and this is sacrum and coccyx labeled here. Then this black line outside, that is the durameter. Then the orange line here is the black line is not on a durameter. Black line is durameter and arachnoid meter together. Okay, black line, durameter and arachnoid meter together. Blue line is the pyometer. Okay, since arachnoid meter is also represented by the black line, this orange line is not a line, actually it's a space, subarachnoid space. Okay, subarachnoid space between the pyometer and arachnoid meter. Then this yellow area is actually the spinal cord. That is the nerve tissue in the spinal cord. Now you can see at third month of intrauterine life in the fetus, you can see the spinal cord extends to the end of the vertebral column almost. Then if you compare this with the child at birth, you can see that the spinal cord growth has fallen short of the growth of the vertebral column. Now the lower end of the spinal cord is at the lower limit of L3 vertebra. Okay. Now the dura mater and arachnoid mater, they are still attached to the sacrum. Pia mater, which was attached to the coccyx, initially all were attached to the coccyx, which extends up to the coccyx, it remains its attachment to the coccyx and the lower part of the sacrum. So you can see it as a line here. Later you will learn this as phylum terminale when you learn the structure of the spinal cord. And the spinal cord is here now. Then in the adult life, if you check the adult spinal cord level and the vertebral column, you can see that the lower limit of the spinal cord lies at L1 vertebra, lower limit of L1 vertebra. This is the adult picture. Now, in both these, in infant, newborn one, and in the adult, you can see this long extension of the subarachnoid space. Long extension of the subarachnoid space. And this is less at birth, but in adults it's much more. Actually, you can, if you want to take the cerebrospinal fluid that fills the subarachnoid space, by now you should know that, you can take CSF by passing a needle between these vertebrae. Usually you go somewhere here, I think, to be safe, because if you go somewhere there, you will damage the spinal cord. Here you cannot, there's nothing for you to damage. So you either go between L3 and L4. If it fails, you can go between L3 and L3, which is not really recommended at the beginning. But in newborns, in the first year of life or something, when you have to do lumbar, you call it lumbar puncture, you have to go between L4 and L5. There is no choice because this is too close, L3 and L4, to the spinal cord. Okay. Then this is called lumbar puncture. I told you, you go between these vertebrae to reach the subarachnoid space. So this is not spinal cord, it's cerebrospinal fluid they have drawn. Okay, don't get confused. Okay. Then developmental anomalies. There are certain developmental anomalies related to the development of the central nervous system and the nervous system as a whole. Now, sometimes during the development, there can be stenosis of the cerebral aqueduct. It doesn't develop properly, there is stenosis, and CSF tends to get accumulated in the area of the brain above that. Okay, so you can see the brain enlarges because of the large ventricular cavities filled with cerebrospinal fluid, and the skull is not well developed at that stage. Therefore, they get separated out at suture lines, the skull bones, and you can see a huge head here compared to the size of the face. You see the size of the head. Sometimes it's operable, sometimes, depending on, I'm not an expert on that. You have to put a shunt, bypassing the stenosis. Sometimes it's put into the peritoneal cavity or somewhere with a tube, but I don't know at which stages they can correct it. Adults, of course, if something develops new, they can correct it, but in small kids, if the brain damage is already there, there's no point in correcting it, I think. But then this microcephaly concept, you can see this here. This person's face is, compared to the face, head is very small. Okay, head is very small because this is again a problem with the development of the brain when the neural tube does not develop in the forebrain vesicle side, because the skull actually develops based on the development of the brain. When the brain does not develop, the vault of the skull does not develop. Okay, so because that's the stimulation for the development of the skull. So because of that, you get the small head area. Okay. Then other neural tube defects, important neural tube defects, are due to the failure of the closure of the neural tube, as I mentioned before. Okay. So this neural tube defect, which is a very hot topic actually, is due to not single factor, it's due to multiple genetic and environmental factors. So many things can cause neural tube defects, some things you never know. Okay, but people have done research, and at least they have found one factor that they can actually prevent, which is an environmental factor. One factor that can prevent the folic acid, because in between these factors, you get large number of mothers who have folic acid deficiency. When they give rise to babies, they get neural tube defects. There are problems with the closure of the neural tube. Actually, after people started giving folic acid during pregnancy, they could prevent about 50 to 70% of the neural tube defects. Okay, but to do that, I told you that neural tube starts very early in the development, even before you know that you're pregnant. At third week, after missing a period, 3 weeks is actually, you don't know you are going to miss a period. Okay, so a lady getting pregnant and having a baby inside 3 weeks old, the next period has not missed. Okay, so then if you are going to wait for you to, if you are you're planning a pregnancy, if you are planning a pregnancy and you are going to use folic acid once you miss a period, it's too late. Neural tube defects will be set in before. So you start folic acid if you are in that age category. Okay, and you are married and you are planning a pregnancy, whether married or not, if you're planning a pregnancy, then you have to take folic acid before. Okay, not before, not after you get pregnant. Okay, and these neural tube defects are common in cephalic region and lumbosacral region. Lumbosacral region is, I think, more common than any other place. So these are a few of the neural tube defects. Now, anencephaly is a failure of the cephalic part of the neural tube to close. So you remember this cranial neuropore. If that does not close, what happens is the cranial neuropore, if it is left open, you know that the cavity of the tube is exposed to the amniotic cavity there. Baby is surrounded by the amniotic cavity and there is amniotic fluid. So if the craniopore is open, okay, amniotic fluid will go into the neural tube cavity, and that will destroy the developing nerve cells. They undergo necrosis, and that actually destroys the brain part completely. And then if the brain part is not there, as I said before, then the vault of the skull also does not develop. You can see it's like, it has been cut by a knife, like, okay, so it's flat. Now these are all, fatal conditions, so you get a stillbirth. Okay, so but the thing is, people don't wait nowadays. The moment you detect this in ultrasound scan during pregnancy, you don't allow the mother to undergo this mental trauma of delivering a dead baby like this. They will terminate the pregnancy. Okay, immediately when you detect it, they will terminate, because even if these babies are born, either they are dead. I think 99% are dead, I think. If for some reason, some live, it's not a useful baby. Then other thing is, you can get this sac-like protrusions from the brain and membranes, covering it, which are called meningoceles. You can have two types of meningoceles, some in the head, some in relation to the spinal cord, in relation to the brain, head and the vertebral column. So you have meningoceles here and meningoceles in the spinal cord as well. We'll come back to it later. So when it comes to this meningoceles in relation to the head, as you can see here, there are three types. I got this from Langman, very nice diagram. There are three types. One is meningocele. One is meningocele, where if you follow the diagram, you have the skin covering outside. Then there's a defect in the skull, through which the meninges herniate with the subarachnoid space. Arachnoid meat is here, you can see. Dura also can be there, I think. And it is filled with subarachnoid cerebrospinal fluid. So that is called meningocele. The brain tissue is intact here inside. Okay, so it's not a very serious condition because the brain tissue is anyway inside. Then the second one is meningomyelocele. Encephalo means the brain. So it's not only meningocele, meningo as well as encephalocele. So you get the dural, with the skin covering, you get the dural, the meningeal coverings. Then you have the subarachnoid space surrounding it with the brain tissue to some extent here. Or you can get, in severe case, you can get the ventricular space also going in which case you can put meningohydroencephalocele. Okay. Meningo means, this meningeal layers. encephalon means the brain, hydro means the ventricular cavities. Okay, but have a rough idea. Okay, not a very extensive knowledge about all these terms. Then this concept of spina bifida, which you need to know, some aspects of it. And it is the basic problem in spina bifida. Is the bifida means, it's divided into two sides, right? The arches, we said the sclerotome of the somite gives rise to the vertebra, and initially it develops from the anterior side, the body, and then, vertebral arches are formed by the migrating cells from the sclerotome. Cells migrate and form the vertebral arches. In this condition, vertebral arches are not formed properly, causing a gap in the spine. Okay, and this is the most common form of neural tube defect. If somebody asks about the commonness of the neural tube defect, it is the spina bifida that is most common, and it's more common in the lumbosacral region. But you can see here the case of spina bifida in relation to the neck area. Here it's not head, it's the neck area, I think, which is common, which is not very common like the lumbosacral area. Okay, so this I already mentioned, and based on which structures herniate, just like in the meningocele of the head, based on the structures that herniate through the defect, it is classified into several forms. So these are the four types of spina bifida: spina bifida occulta, meningocele. You remember there was a meningocele in relation to the brain also. Then myelomeningocele and rachichisis. We call it rachichisis. Okay. So here, in relation to the brain, you have meningoencephalocele. Okay, here you have meningomyelocele or myelomeningocele, either way. Okay, meningomyelo means the spinal cord. Okay, so if you, okay, we'll take one by one. Now, spina bifida occulta. Spina bifida occulta, the first one, occulta means it's not seen outside. We can also call it hidden spina bifida. Hidden spina bifida. Okay, by spina bifida is not seen outside. You cannot detect it. It's the most mild form. It has no effect on the neurological system. Only thing is you can have, some unstable features of the spine and all. You might have to correct it. Sometimes this is noticed by having some feature in relation to the defect. Now you can have a small birthmark, like in this baby. You have a small birthmark outside that actually indicates that there can be spina bifida occulta. Okay. Sometimes there can be a dimple like this in that area, or there can be some hair growth in that area like this. Okay. Or sometimes you don't have any of these, but some small fat pad in that area, collection of fat underneath the skin, which will indicate the underlying problem. But anyway, this is not a very serious problem. This is the mildest form of spina bifida. Then the meningocele, just like in relation to the brain, you saw the meningocele, you get the skin covering and the dural, the dural and arachnoid covering here with the extension of the subarachnoid space filled with cerebrospinal fluid. Then the meningomyelocele or myelomeningocele, whatever you call it, you get the spinal cord coming into the cystic lesion, which is a little bit of a serious condition, because that can have problems actually, because there can be some certain compressions and paralysis or weakness of lower part below this part. Okay. Then the rachichisis, I think most of the babies born with rachichisis are dead actually. Only very few cases have lived for few days, I think. About 6 or 7 cases when I checked the internet. But usually it's death, stillbirths. Okay. Here what happens is the neural tubes, they don't close, they remain open. Okay, just like in, I said the cranial neuropore is kept open in anencephaly, and that destroys the nervous tissue because of the amniotic fluid going in. Same thing can happen here, so it destroys the neural tissue. I think neural tube remains open. Maybe this is in relation to the caudal neuropore and all that. Okay. So then, so by far, this is very uncommon as well as, very lethal, deadly condition.